Hey there, micro friends. Welcome to another episode of Let's Talk Micro. As always, I am so happy that you're here with me. And if this is the first time you're listening to Let's Talk Micro, well, thank you for choosing it as one of your podcasts. So, last week, we started a series on gram negative cocci. And those of you that are longtime listeners, you know that early on we did a couple of episodes. They were a little general. One was on like an overview of gram negative cocci, and we did one on the catarrhalis disc. Now, given the fact that organisms like Neisseria gonorrhoeae or Neisseria meningitidis, they're so crucial and so important for us to make sure that we correctly report and we are able to detect in cultures and other microbiology tests, I thought it would be a good time to do a more comprehensive series about gram negative cocci. Those of you that work in the lab, do you know that sometimes reporting a gram negative cocci can be a little scaring or even daunting, right? Maybe you're that tech that finds themselves alone in the lab and you have maybe a blood culture or a cerebrospinal fluid and you think it's a gram negative cocci, but you're not sure. And maybe you're afraid that you might be missing something like an Acinetobacter that has those plump coccobacilli and you want to make sure that you don't report the wrong thing, right? That affects your patient. So it can be a little scary sometimes. And that is the goal of this series is to reinforce those concepts so even students and those of us that work already in the field in the bench that to make us a little bit stronger so we are able to rule some things in, rule some things out, and be more comfortable when we're working in the lab with gram negative cocci. Now, I know that, of course, molecular testing, it keeps becoming more and more important, a larger part of what we do in the lab. And by now, most labs, I think that at least from medium to large size labs, they might have an instrument to run PCR or other molecular tests on blood cultures or CSF. But maybe you're in the lab and you don't have one of those instruments. I always advise, if unsure, when you have these tests, these instruments, these molecular tests, wait for the result if you have it, and also, consult with someone if unsure of what you have either on the plate or on the gram stain. So on the last episode, we started all the way from the basics, describing the gram stain, the morphology. We talked about the different genera and species of gram-negative cocci. We talk about decolorization issues and places where you typically see these species. And by that I mean that, of course, if you have a blood culture or a cerebrospinal fluid or a body fluid, you if you see something that you think is a gram-negative cocci, you definitely are going to be more afraid to report it. But let's say that you are looking at a sputum culture. Well, in that particular sample, you should feel okay when you see a gram-negative cocci and you report that. Because as you know, non-pathogenic Neisseria, it's part of the respiratory flora. So you will see gram-negative cocci in that particular source. And when I mentioned, decolorization issues, one thing is with Neisseria meningitidis that it can resist being decolorized because of capsules and pili. And this decolorization issue is something that we also see very classic with Acinetobacter baumannii. So that's something that we have to keep in mind because, Acinetobacter baumannii, you will see sometimes that it retains that purple in the gram stain. And as I mentioned before, sometimes, you can confuse it with a gram-negative cocci, but I always advise you to scan the slide. And even though you see these plump coccobacilli, if you look around, you're going to see some that are rod shaped. So really look for that. It's not going to be thin rods, they're going to be like thick bacilli, but scan around. That does not happen when you have Neisseria meningitidis. So keep that in mind. And I said this in the last episode, but I invite you to view in the lab. Go ahead and make a gram stain of Acinetobacter, one of Moraxella or Neisseria gonorrhoeae, and one of Haemophilus. And you make those slides and then compare them and see, watch out for all the differences. Compare, the size of a coccobacilli like Haemophilus, where you cannot tell what it is. Compare that to the kidney shape, the kidney or coffee bean shape of the Neisseria, and those plump coccobacilli of Acinetobacter. I definitely invite you to do that. Okay, so in today's episode, we're going to focus on media and biochemicals, more specifically over Neisseria and Moraxella catarrhalis. And in the previous episode, we mentioned other species of gram-negative cocci, and we talk about Veillonella. And when it comes to it, there's not much to say because, it is an anaerobe, a gram-negative cocci, and it will grow on anaerobic media that it's meant for gram-positive organisms. So it will grow on a blood plate, blood agar plate incubated anaerobically. It will grow on on a CDC plate. So if you have media that is for anaerobes, where typically gram-positive anaerobes grow on, Veillonella will grow on it. So we're going to focus our time on Neisseria and Moraxella catarrhalis. Now, in the audience, we have students, we have people that are preparing to take the national certification or some sort of certification to be able to work in the lab, to be licensed and be certified. So I'm going to spend a little time talking about some things that you might see when you're taking that test, that certification test. And I'm going to start talking first about Neisseria gonorrhoeae. It grows in a carbon dioxide, CO2, environment. And as you know, in the lab, we have CO2 incubators and non CO2 incubators. And typically, maybe depending on how your setup is, on the non CO2 incubators, some people keep like the urine cultures and things like that. And in the CO2 incubators, they keep the other types of cultures. Typically, if it has chocolate, if it has modified Thayer-Martin, Martin-Lewis, those plates, they go in the CO2 incubator. So let's spend a little bit of time talking about collection and transport because it might help you if you're taking the test, the certification test. If Neisseria gonorrhoeae needs to be ruled out for the collection, the recommendation is to use rayon, Dacron, and nylon tip swabs, one of those three. As far as transport media, amines media or a liquid transport media is recommended. And it is definitely recommended for the sample to be transported to the lab as soon as possible. And even though there are studies that show that after refrigeration, Neisseria gonorrhoeae has still been recovered after 24 hours, it is still the recommendation to plate it as soon as possible. And the reason that I wanted to mention this is because I want to talk about a transport system. And those of you that have taken the certification and those of you that are studying, to take maybe something like the ASCP or AMT, and you know that something that helps us study and we do is, we make these connections and we get case studies and we look for classic things in the question that helps us with the choices, right? Things like long thin rods for lactobacillus or hockey puck for Moraxella catarrhalis. So you might hear something called JEMBEC, which is JEMBEC, on your test. You might have a question about that, so let's go ahead and talk about this. This system consists of a modified Thayer-Martin plate, and I will talk about that, a tablet that generates CO2, and a plastic bag. The tablet is composed of sodium bicarbonate and citric acid. Very simple, the setup. You inoculate the plate, and you place the tablet in a well that the plate has, and then place the plate in the bag. And what happens is that moisture activates the tablet, which generates CO2. And then you transport this to the lab and incubate it at 37 degrees in a CO2 incubator. And something to note is that this system is also recommended for Neisseria meningitidis. And of course, those of you that work in a lab, you know that sometimes, microbiology is more centralized and maybe there's one hospital that serves as the micro lab for many. Maybe the one that you work in, you don't do cultures, like you don't set them up, you don't read them. So sometimes, there might be a delay in your sample being transported to that microbiology lab. And this is what the system is for, right? If the sample is going to take longer than 24 hours, it gets collected in this and it should be transported as soon as possible. But even if there are delays in the system, at least you're providing the right conditions, from the beginning. You're giving that organism the plate, the media that it needs, the conditions like CO2, and at least you give it a chance. And if you work in a lab, there's no question that Neisseria gonorrhoeae needs CO2, especially those of you that do quality control. And sometimes, your monthly bugs or some things like that, you sub them to like blood agar And once they grow, you put them in the fridge. And then you pull them out a few days later, you sow them again, and they still grow. But Neisseria gonorrhoeae is not like that. You take that plate out of the incubator and you put it in the fridge. You have a very short time before it dies. And I'm sure you know this if you work in a micro lab. Pretty much everyone has like the gonorrhea, the Neisseria gonorrhoeae QC plate. You keep one in the incubator. And even then, if you keep it after a few days, sometimes, it still dies as well. Okay, so let's continue going. So we have gone over the gram stain. And as you know, with the gram stain, you have media. So let's go ahead and talk about media. Your standard setup for a culture, right? It has blood agar, chocolate, and McConkey. And by now, all of you should be familiar with it. And if you need a refresher, there are some episodes in our catalog where we talk about media, so I invite you to check those out. So you have that standard setup test, and you know that typically if your source is genital, then you will add a modified Thayer-Martin. If you're doing like a Neisseria gonorrhea culture, you will do like a chocolate agar and a modified Thayer-Martin plate. But overall, most of these Neisseria species and Moraxella catarrhalis, they do grow on blood agar and chocolate agar. And you might find some references where it says that Neisseria gonorrhea does not grow on blood agar, but it does. It actually does, but not as strong as it does on chocolate agar. When it grows in blood agar, you will see like smaller colonies. Sometimes, you think that the plate is too young. And this is something very important for techs out there because this is something that I have experienced. I haven't done it, but I have seen it in the lab, and that is not good for patient care, which is when you are working on urine cultures. The standard setup for urine cultures is blood and McConkey. You don't have a chocolate plate. And I have seen scenarios where people are working on urine culture, they look at your McConkey, there's no growth, they look at the blood agar, and they see the small colonies, they think the plate is too young. They assume it's a gram-positive cocci that is too young. They say too young, and they reincubate the plate. And then the next tech comes in, and they look at the plates and say, well, it still looks too young, let's reincubate. And sometimes with that too young reincubate, that plate spends 2 or 3 days in the incubator. And that is not good microbiology. It's very irresponsible. And the one thing that I have seen is that after those 3 days, no one has even done a gram stain, which is such a valuable and basic tool. So keep in mind, you can get Neisseria gonorrhoeae in your urine cultures. It will grow in blood, but not as strong. And as a general rule, on blood plate, any small colonies, small to pinpoint, they can be anything from gram-positive rods to yeast to gram-negative cocci. So please, if you have this presentation, do a gram stain. And if you see something that looks like gram-negative cocci, check the patient's history. Typically, you see a PCR done for Neisseria gonorrhoeae there that is going to be positive. And then go ahead and take those colonies and sub them to a chocolate plate, to a modified Thayer-Martin plate, and go from there. Which brings me to the modified Thayer-Martin plate. I keep mentioning it, so let's go ahead and talk about it. So this is an enriched and selective medium for the isolation of Neisseria gonorrhoeae and Neisseria meningitidis. The enrichment portion is chocolate agar, and the selective portion is antibiotics. So it has colistin, which inhibits most gram-negative bacteria, vancomycin, which inhibits gram-positive bacteria. It has nystatin, which inhibits yeast, and it has trimethoprim, which inhibits the swarming of proteus. And speaking of proteus, maybe some of you are thinking, well, proteus is resistant to colistin, and well, you have trimethoprim there. So even if it grows in the medium, it's not gonna swarm. Keep in mind that if you incubate that plate for too long, eventually you might see some swarming there. Something to mention is that, as you know, these bugs, they don't go to school, they do what they want, and also you know the components of the medium. So even though this plate is intended for the isolation of Neisseria gonorrhoeae and Neisseria meningitidis, other organisms can grow on it. So having growth does not automatically mean that you have Neisseria, gonorrhoeae, or meningitis. If you see growth there, it should be followed by a gram stain to see what you have. And even if it's gram-negative cocci, you still have to do further testing because Neisseria lactamica and Moraxella catarrhalis, they do grow on modified Thayer-Martin. And something that I found to be useful, and it's always good, as I said, to learn the components of your media. But in the lab, sometimes, we have myroides and proteus together. And it's very hard to isolate the myroides because the proteus keeps swarming. So one thing that might help you is that suspect colony to a modified Thayer-Martin, incubate it, and maybe after 10 hours or so, when you see growth, sub it to another medium. You might keep the proteus, you might hold it back long enough for you to get that myroides out. So that's something that I wanted to share with you, something that I have done. It has worked. So keep that in mind. It might not work for you, but still it's something good to have in mind. Okay, so then you have Martin-Lewis, which is a modification of modified Thayer-Martin. And in this agar, instead of nystatin, there is anisomycin for yeast, and there is also a higher concentration of vancomycin. And this plate is intended for the selective isolation of Neisseria gonorrhoeae. There is also another medium, which this one you might see on your certification exam questions. It's called the NYC agar, or New York City medium. It has lice horse blood, horse plasma, and the same antibiotics as modified Thayer-Martin. And according to Bailey and Scott's diagnostic microbiology, we're getting technical, this medium has an advantage, which is that grows organisms such as Uroplasma urolithicum and Mycoplasma hominis. And there's a test that I want to go over. And of course, nowadays we all have Vitek, Moldy, at least, maybe in the US and maybe Europe. I know some places they still do a lot of manual tests, but we still have to learn them. We have to, we get questions about them and, in certification exams. So we have to learn them. So we learn them in school. And this test is called the ONPG test. And the purpose of this test, it's to differentiate, lactose fermenters from non-fermenters when it comes to Enterobacterales. But this test, it can be used to differentiate between Neisseria lactamica and Neisseria meningitidis. Lactamica is ONG positive, and Neisseria meningitidis is negative. So the ONPG test, which is the O-nitrophenol beta D galactopyranosidase test, is used to detect the enzyme beta-galactosidase. In this test, the ONPG substrate is hydrolyzed by beta-galactosidase to produce galactose and O-nitrophenol, which results in a yellow color. To perform the test, you have an inoculum of the organism, and you add the ONPG reagent, which is typically in the form of a disc or a broth, and you incubate it at 35 to 37 degrees in ambient air for 4 hours. The test is then observed for a color change. A yellow color indicates a positive result, while no color change indicates a negative result. So keep that in mind, and thank you for bearing with me as I pronounce that whole name. I appreciate it. So let's talk a little bit about biochemicals. As far as biochemicals, Neisseria and Moraxella catarrhalis, they are oxidase positive. And one test that we talk about and we learn in school, which I have mentioned before, it's the acid detection test, also known as the carbohydrate utilization testing. And this test uses cysteine trypticase agar, CTA, with 1% of a specific carbohydrate, such as glucose, maltose, lactose, or sucrose, and it uses phenol red as a pH indicator. When acid is produced due to fermentation, the medium changes from red to yellow. And different Neisseria species, they produce different patterns. They ferment different sugars. For example, Neisseria gonorrhoeae ferments glucose. Neisseria meningitidis utilizes glucose and maltose. Neisseria lactamica utilizes glucose, maltose, and lactose. And a fun way of learning that is that, starting with gonorrhea, you associate the letter of the species with the sugar. and you keep going. For example, Neisseria, gonorrhoeae, G, glucose. The meningitidis is the M, so you start with glucose and then add maltose. Lactamica, L, glucose, maltose, and lactose. So that's a fun way to remember, and you associate the letter of the species with the carbohydrate they utilize. One thing to note is that this test is no longer recommended for primary identification, and this is because you can have some misleading results when you have Neisseria species that are oxidative rather than fermentative. And as you know, overall, accurate identification is essential, right? You want to make sure that you identify the correct organism because, depending on the organism, your treatment choices change. So you want to make sure that you have an accurate identification. But when it comes to Neisseria gonorrhoeae, it's very important because there can be some legal consequences, right, by identifying this organism. So keep that in mind. Okay, so when it comes to other testing, let's talk a little bit about Moraxella catarrhalis. So there's a test called the butyrate disc test, also known as the catarrhalis disc. And this test detects the enzyme butyrate esterase, and it aids in the identification of Moraxella catarrhalis. So let's get a little bit technical. Moraxella catarrhalis has an enzyme called butyrate esterase, which releases indoxyl from indoxyl butyrate. This produces an indigo color in the presence of oxygen. And this is a very simple test. You get the disc and you place it on a slide, then apply your colonies using a wooden stick. A positive result is a blue color developed within 2 minutes. A negative result is no color change. Keep in mind that you have to make sure that what you place on the disc it's a gram-negative Cocci. Otherwise, you can get some false positive results. And this brings me again to if unsure, you always do a gram stain. Of course, as you become more experienced, you see some organisms and you already know what they are. You have an idea. You get proficient enough that you can do that. And Moraxella catarrhalis has that hockey puck where you touch the colonies and the slide around the agar. But if you're starting to work with the organisms, and even if you're experienced, a gram stain is always great practice. So bottom line, if you're doing this test, make sure that what you have is a gram-negative Cocci. Now, why is this test useful? Well, it lets you presumptively identify Moraxella catarrhalis. You have the colonies, you see the sliding, gram stain is gram-negative diplococci, you do the test, you have a positive result, you can go ahead and call it Moraxella catarrhalis. You don't need to do anything else. You don't have to put it on the Vitek. You don't have to do Molotov. So once you identify it, you're done at that point in time, and you can move on to the next culture. As you know, the microlab, you have a lot of cultures, you have a lot of samples. You have to be conscious of your time. You don't want to be wasteful. So if you have tests that fully identify the organism and you don't have to wait for things like Molotov and Vitek, take advantage of that. You identify it, you move to the next culture. Okay, so let's do a quick recap. By now, on the previous episode, we talked about gram stains. Now we are talking about media. So we talk about the JEMBEC system, the transport system. We mentioned the fact that most of these, like Neisserias, they grow on blood and chocolate. Keep it in mind that Neisseria gonorrhoeae, it grows on blood, but very weakly. So make sure you don't miss that on the urine bench. And then we talked about Modified Thayer-Martin, Martin-Lewis, and NYC agar. These are selective media. We also talked about the ONPG test that can help differentiating between Neisseria meningitidis and lactamica. And we talked about the carbohydrate utilization test. And then we talked about the catarrhalis disc. So we have all these tests that are manual, and we have to learn them in school, right? Many parts of the world, they still do them a lot heavily. But let's go ahead and in the next episode talk about modern approaches to testing. And of course, we have things like MALDI, we have Vitek, and then there are nucleic acid tests and molecular tests. So let's talk about those on the next episode. Okay, friends, that was the episode. I hope you enjoyed it. As always, I enjoy sharing this information with you. And of course, if you want to find us on social media, we are on Instagram, TikTok, YouTube, and Blue Sky Social as Let's Talk Micro, X as Let's Talk Micro 1, LinkedIn as Luis Plaza. There's an email address, which is letstalkmicro@outlook.com. There's the website, letstalkmicro.com. I invite you to subscribe to the website. There are study tools, episode updates, licensing information, and more. So either via the website or social media, you can reach out with any feedback, any topic suggestions. And of course, there's always the question of sometimes, people ask for the link to the episode, but you can find us on any podcast platform. So if you go to Apple, Spotify, Amazon Music, Good Pods, we're right there. So I invite you to subscribe, download episodes, and if the app allows you to do so, please leave a review. Thank you so much for the support. And speaking of support, if you want to support us in any way, you can do so via Venmo or via Buy Me a Coffee, KOFI, both links available on the show notes. As microbiologists or infectious disease specialists or pharmacists, definitely learning about antibiotics can be difficult. So if you're looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. And as always, these are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. The Let's Talk Micro podcast is supported by the American Society for Microbiology. To learn more about microbes, visit asm.org. And that, micro friends, it's the end of this episode. I hope you enjoy learning about media and tests for gram-negative cocci. As always, I enjoy sharing this information with you. As always, thank you for the support. I'm so grateful. Stay tuned. Great things coming your way. Are you thinking about attending Microbe? Hey, I'll see you there. But as always, continue bringing that passion to what you do. It's so important. You do such great work. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.